Welcome back to the AP Biology Bros podcast, broadcasting live from the nucleus, where the DNA is stable and the drama is unstable.
Yeah.
I'm Gray, your host, aka DNA. Genetics are stored inside the cell, and I control those instructions. I don't leave the cell for security reasons, you know what I mean? But without me, there is no show.
Yeah. Well, howdy everyone. I'm Gage, RNA polymerase. Well, I'm the one who binds the promoter regions and starts transcription. If you need someone to get things done, I'm the cowboy to do it.
I'm Amir, messenger RNA. I carry the genetic instructions out of the nucleus. I get capped, tailed, spliced, basically edited, and then I deliver the message.
Yeah, I'm Preston, the ribosome. I read mRNA and I build proteins. Amino acids, I connect them. Peptide bonds, that's my specialty.
Today's episode, how cells produce proteins and how they avoid complete biological chaos.
Sponsored unofficially by mitochondria.
Yo!
Still the powerhouse.
After that break from our sponsors, let's start at the beginning, transcription. Since we're in a eukaryotic cell, DNA never leaves the nucleus.
Correct. So I bind to the promoter region of a gene. I separate the DNA strands and read the template strands to form RNA.
Remember guys, RNA uses uracil instead of thymine.
And that's where I come in. I'm built using complementary base pairing, the coolest thing around.
Before mRNA leaves the cell, processing must occur.
5 cap added, poly A tail attached, and introns removed during RNA splicing. Now, only exons remain.
That's facts. So basically, quality control before shipping.
I guess, exactly. Well, now I'll see you guys in the ribosome. Peace out.
There you go.
Notice something here, listeners. Transcription happens in the nucleus. Translation happens later on in the cytoplasm. Separation of process. That's a key eukaryotic feature.
Cool.
Ribosome break. Ribosome break.
Now we move to translation. Preston, walk us through it.
mRNA.
And I got you. I'll tell you about translation. Translation begins at the start codon. AUG. Because that's where we met, though, eh?
I see what you did there. I see what you
like
did
the
there.
amino acid? Yeah. Alright, now walk through it.
I'm gonna go through the ribosome.
Each codon matches with the tRNA anticodon. tRNA delivers the correct amino acid, and peptide bonds make a polypeptide chain.
Translation continues until the stop codon is reached.
Now this represents central dogma, which is DNA, RNA, and protein.
Transition!
Go. Alrighty, time for our debate segment. Question today, which process is most important?
Ooh.
Oh,
Well, transcription.
Yeah.
Without my function, there is no message.
Yo, nah. It's always translation. Without my process, there's no functional protein.
What are you guys talking about? It's obviously regulation. Without control, genes would be expressed non-stop.
Well, technically I'm the blueprint around here.
You don't even leave the nucleus, bruh.
Basically a high protected document.
Okay, and?
Let's talk mistakes. Who caused the last frame shift?
Allegedly.
You inserted an extra nucleotide.
That shifts the reading frame and changes all downstream codons.
And which alters every amino acid after that point.
Okay, fine. That one was bad. Chill.
So perhaps we all matter?
Fine. The translation still does the heavy lifting.
Yo, you hear that lysosome got frame mod?
Yeah.
Ribosome
So,
factor.
it's actually terrible, bro.
That was me. I'm not joking.
Breaking news, guys.
What?
Listeners, there's a mutation detected.
Are you serious?
I swear I only inserted one extra nucleotide. I promise. Like,
That's a friendship mutation!
no.
Oh my god.
All codons after the mutation change!
I'm genetic chaos!
No, this form of protein detected. Possible loss of function.
This could contribute to disease!
No! New studio here because of Preston and his mutation. Let's talk about prokaryotic regulation. Amir?
Sure, let's talk about it. Oh, in prokaryotes, genes are often expressed as operons, like the lac operon. I'm a repressor protein. Gene off.
If lactose is present, an inducer will bind to the repressor.
The repressor then detaches, and the gene is now expressed as on.
That's efficient control. Genes are only expressed when they are needed.
And that's prokaryotic regulation.
Quick meme break. Time flies when you're having a good time at AP Bio.
Exactly, bro.
The unit's gone by so
Why the unit's
fast.
going by so quickly. We're learning and we're engaging, this project. It's really helping us, develop our understanding on the unit. Prokaryotic regulation, transcription, translation, the genetic dogma, it's all
does
expanding our
elongation
knowledge.
mean?
Huh?
What is
Oh, we
elongation?
don't.
So, let's talk about eukaryotic regulation now. Eukaryotes regulate gene expression differently. DNA wraps around histones, forming chromatin. When chromatin loosens, transcription factors can bind.
Enhancers increase transcription. Silencers decrease it.
So even access to DNA is regulated.
Exactly.
That's what I was
We're
thinking.
not thinking like that, though.
Yeah.
Before we close, let's summarize here.
Well, first off, we lost our podcasting room to mutation monster. And for our main points, transcription copies DNA to RNA.
Translation uses RNA to build proteins.
Mutations can alter protein structure and function.
Regulation controls when genes are expressed.
DNA to RNA to proteins. Now join us next week for ATP. Underpaid and overworked.
If lactose is present, an inducer binds to the repressor. Efficient control over here.
That's efficient control. If I haven't never seen one. Yeah, I lost my mind.
If I have never seen
tRNA delivers the correct amino acid and peptide bonds create a polypeptide chain.